I have the pleasure of introducing our next speaker, Dr. Gulbu Uzel, with the National Institutes of Health, with the National Institute of Allergy and Infectious Disease at the NIH in Bethesda, Maryland. Thank you.
Thank you. So my talk comes timely after your introduction to some genetic counseling and having heard about whole exome sequencing, especially now being commercially available. So I'm going to tell you about the stories of discovery, 3 genetic discoveries leading to immune dysregulations. They are, in a way, immunodeficiencies. There are some immunodeficiency component to it, but they are more of defects of immune homeostasis. So thinking about immune homeostasis, it is our lymphocytes, when they encounter the antigen, they proliferate and expand. That's what the antigen response is. And some of them become memory cells. I see that there's a PCMAC conversion problem. Some of them become memory cells. Rest have to die, so that this whole cycle keeps going on, because we cannot keep expanding our lymphocytes. On the other hand, immune tolerance is very complementary to immune homeostasis, where a self-reactive T and B cells should be removed. We all start with some self-reactive T and B cells in the thymus and the bone marrow. They need to be removed. This whole process of deletion, as well as removal of memory cells, is done by this process called apoptosis, programmed cell death. So defects in apoptosis may lead to, of course, defects in immune homeostasis and tolerance. So there are known immune defects of apoptosis. You've heard about ALPS, autoimmune lymphoproliferative syndrome. Mike talked about it, where these patients have big spleens, they have cytopenias, big lymph nodes, and they may develop lymphoma. And there are genes that are associated with it. We are now all into genes that lead to these clinical syndromes, because the gene that's affected, or the mutation, specific mutation, may even dictate the outcome or the phenotype, or how well or how bad the patient will do. And so these are the known genes. There are some newer ones that have been discovered. These are gene discoveries, again, within the last 10 years, like NRAS, KRAS, caspase-8, FADD8, FAD. And I'm just fleshing these names because we don't have time to go into these, but they're really interesting, challenging defects, diseases. And the one that I'm going to tell you about is this last one, called protein kinase C delta defect. So in around 2010, I saw a patient in the PID clinic at NIH, was referred to me from Chicago, and this patient had extensive lymph nodes. You can see there, axillary lymph nodes, a huge liver, huge spleen. He even had cava superior syndrome because of the mediastinal lymph nodes compressing on the cava superior. And otherwise, he didn't have too many infections, maybe a ear infection or lymph node infection, but this was very impressive. So when we see this kind of picture, extensive lymphadenopathy, lymphoproliferation, one of the genes that's checked is genes that lead to ALPS. And he came to us after all those mutations being done commercially, commercial labs. And then we thought, well, this looks like ALPS, but it's not really ALPS. There's no mutation. And we looked at his flow cytometry a little bit in more detail. And you can see here what I highlighted are the B cells, CD20 positive B cells. He had an increase in the number of total B cells, but also he had these more immature B cells. So that was a different type of ALPS, we thought, like B cell defect in apoptosis. So then we went back to look at his lymph nodes, because if you are talking about B cells, you better look at the lymph nodes. He had a lot of lymph node biopsies in his lifetime, 4 or 5 of them. And as you can see here, you're looking at the normal, and on the left-hand side, and patient on the right, comparing the lymph nodes. normal has a very nice germinal center, the follicle that you're seeing there, with a nice cuff around it. In the patient, that cuff has disappeared. The germinal center is expanding. That's what it looks like. And then the stains that you're going to look at down below reflect how reactive or organized the lymph follicle is, and how well the T cells are talking to B cells. So it's a very disorganized, very active lymph node with not so good T cell, B cell coordination. So we did whole exome sequencing with a project. It was a little venture that we had with a colleague of ours at Clinical Center, Bosco, who left U.S. for Brazil recently. But we found a candidate gene, and these are genes that have not been associated with disease before, so very different than what you get from gene DX. So found the homozygous point mutation in this gene PRKCD or PKC delta, and this missense mutation, it's not causing a stop or causing a out of frame shift, but this missense mutation affected the signaling section of the protein that leads it to the nucleus. So it's a nuclear signaling sequence, nuclear localization sequence that was altered. And we have done multiple in vitro studies that we show that that was what was affecting his problem. So as you can see, this is the patient. Both parents are heterozygous, so are all the siblings. So there was no history of consanguinity, but both parents were from a small town, small village in northern Mexico. So now we are getting into a small genetic pool and not being related to, that you know of, but actually being related to your husband. And genetically, I mean. So what I showed here is the sequencing and showing that patient has a single signal that is red, and every other member of the family has two peaks, two different colors. So I was going to talk about a little bit briefly about whole exome versus whole genome, but that was done very nicely, elegantly by the speaker before. Basically, what we are looking at by whole exome is maybe 1 to 2% of the whole human genome, but that 1 to 2% is what really is will be where the mutations will be more likely found at. And then we verified this. So then you go back, what you find, you verify the defective protein or protein expression by Western blot or histopathology. And when we stained his lymph node, you can, as you are seeing, looking at the K, where patient lymph node didn't stain for this protein that was as a result of the mutation in the gene encoding for that. And although patient didn't express the protein, his mRNA levels were normal to high, depending on where you looked at. And that we figured out that it was a post-translational problem. And so this protein, it turns out that protein kinase C delta is very important in apoptosis, pro-apoptotic, so helping cells die properly and helping with autophagy, cells being able to digest things that they have to engulf, and helping with cell cycle. So when it is mutated, it is causing these big lymph nodes, but also leading to this abnormal B cell phenotype. Because B cell, it's important for B cell apoptosis, not for T cell apoptosis. So we have a lymphoproliferative defect affecting the B cells primarily. And we did this, we showed this, and when we looked at patients' B cell line, now we are looking at the B cell lines. We take patients' B lymphocytes and transform them with EBV virus, then make them immortal, and we look at their ability of these cells to proliferate, die, or survive in culture. As you can see here in the on the left-hand panel, the white little circles, it's normal controls EBV cell lines. They die much more readily than the patient. But when the patient's cells were cultured in rapamycin, it's a Rapalog, one of the immunomodulators we use today, the apoptosis defect has significantly improved. So when you put this patient on rapamycin that you're seeing in the CT scans below, his spleen and liver significantly reduced in size, his lymph nodes disappear, vena cava superior syndrome improved, and patient has been in good condition, doing very well since then. So we have published this at the same time with this German group. They found a similar problem, lymphoproliferation problem with the B cells, but one of the siblings had significant amount of autoantibodies and developed lupus in that patient. And then 6 months later, a French group has published this family where they had described familial lupus. And they found the mutation, again, homozygous mutation in the same gene. So now what we are looking at is a gene that is involved in deletion of autoreactive B cells. And these are the cells that secrete autoantibodies directed to self. This is what we saw in a patient with lymphadenopathy, cytopenias, or he had autoantibodies, although he didn't have the lupus-like picture, and EBV virus constantly in circulating EBV viremia at low titers. And that's because of a natural killer defect, too. So this disease was a great model for us because it mimicked the knockout mice model. So we get lucky when we find a gene in whole exome through sequencing. And if there is a model, a mice model already out there, somebody has made that mouse in the gene that we find a mutation, then we kind of compare notes, look at the mice. Does our patient have what they saw in the mice? So that was the case, and the patient mimicked the mice exactly, and we were lucky that it worked out this way. But again, this is the problem of describing a new genetic defect with one family. We were more concentrating on lymphoproliferation, ALPS-like phenotype, whereas the other group had a family with lupus, autoimmunity. So their conclusion was, well, this mutation leads to lupus. But I think that the defect in basically explains for both. And we were looking at, we had this project, nice project going on, and we were looking at this patient trying to figure out PRKCD exactly. We found, we realized that we had another candidate gene in a different group of patients with lymphoproliferation, yet different set of problems, different type of lymphoproliferation. So this was not the defect in apoptosis. The apoptosis was intact in these patients that I'm going to tell you about. It was the expansion, accelerated expansion of lymphocytes, and then these lymphocytes skewing away from the naive type of T cells, because you have to keep a certain population of naive T cells, but the patient moved away from being naive very quickly, very rapidly, and became effector memory T cells. So this is a very interesting disease and very, and it's not very rare. We have been seeing a lot of patients with this. So these patients all had recurrent pneumopulmonary infections, bronchiectasis at an early age, very young age. They had chronic lymphadenopathy, big liver, big spleen. They had what is called lymphonodular hyperplasia. So when they had their endoscopies, bronchoscopy or endoscopy, their mucosa had these little bumps, nodules sticking out, which were all reactive proliferating lymphocytes. And then they had recurrent herpes virus infection. They were CMV viremic, EBV viremic. They had HSV, zoster at a young age. So, and they also had autoimmune cytopenias, ITP. So some of the patients had their spleens removed because of hard to treat ITP, and they were hypogam. So this would be a some of these patients could have been were followed actually as a common variable immunodeficiency patient. So when we did the whole exome sequencing, we were looking at genes that would lead to loss of function. But when we found hits in the same gene in several patients that we aligned with the same clinical phenotype, we realized that could have been easily a gain of function mutation. So when we zoomed into this gene and then the protein that it encodes, it's the PI3 kinase 110 delta, it turns out that it is a very fundamental, important enzyme, kinase and enzyme at the same time that processes the lipids in the membrane that helps signal go through, lets other kinases stick to come to the membrane. So we had found 3 different mutations in this gene with the patients with the similar phenotype, although there's a good variety of clinical variation. And it was it's the what you're looking at is the lighter color, gold color is the protein that's mutated. When the mutation happens, it's in heterozygous, only in one allele, one copy, you have the mutations. The P85 is another subunit, doesn't stick to it. When it doesn't stick to it, this 110 delta gets overactivated and does all the unpleasant things that we see in the patient. We showed that the downstream signaling proteins were overphosphorylated, overactivated. And as you can see there, A blue is the patient in every cell we looked at. And we even saw this in not just T cells, B cells too. And we were able to observe this compare the amount of activation in cells intracellularly in the patients with different clinical presentations and try to kind of correlate. Can we understand based on how their cells behave in vitro and translate to what we see in the clinic? So it's the fun part comes here when you look at these pathways, signaling pathways after you find the genetic defect. You try to understand, to figure out how can I intervene with this signal? How can I overcome this overworking system? And we were lucky in a way because this signal went through what we called mTOR pathway. And we have this inhibitor of mTOR, a repalog, rapamycin, or sirolimus or everolimus, which has been used for solid organ transplants. And we thought, out of desperation, let's try this. And we tried it, and patients had an incredible response because you are overcoming an overworking pathway. Patients' cells were able to stop proliferating and expanding. This mutation leads to not just increase in proliferation and change from naive to memory phenotype, but also affects the biochemistry of the cell. When you have a when you have a cell that's going through the mTOR signaling too much, you shy away from the glycolytic pathway, but go to more storing and conserving energy, by that way becoming more senescent. So it's a disease causing old lymphocytes, leading to old lymphocytes. We call them senescent T cells. And senescent T cells are not good because they don't do what they're supposed to do. They cannot kill the viruses. They may recognize the virally infected cells, but they cannot kill them. That's what we showed. We showed that these cells had the machinery to recognize EBV-infected B cells. This is what you're looking at on the top panels is the patient's CD8 T cells. That this is what's called a tetramer staining, is able to go bind the B cells that are expressing different B cell antigens. So we were able to see that it's not a recognition problem or antigen presentation. It's actually the machinery of the CD8 cells be able to kill the infected cells or tissues. And then we did some additional work trying to characterize the kind of cells that are causing problem, why they are so heavy, they are not making as much the right cytokines doing what they're supposed to do. And then we came up with this phenotype that is their T cells are senescent and over shifting from to senescent effector memory phenotype from naive, but their B cells are not normal either. They have a loss of memory B cells, as you can see there, PI3 kinase mutant. This doesn't have this population here, but expansion or expanding transitional cells, which are not supposed to be in the peripheral blood circulation as much in that quantity. So premature B cells are leaking out from the bone marrow, and they do have low immunoglobulins, as you can see here. This is the low normal level for the for IgA for different ages, as you can see here, and then IgM and IgG. Most patients have either progressive hypogammaglobulinemia or they have high IgM and low IgG, which we call hyper IgM phenotype. And we did further studies for looking at B cells, what is the real defect. It's not now stopping at what's going on with B cells, but trying to understand what this protein, this kinase does to the B cells. And we figured out that it's a maturation defect. And B cells don't class switch, don't make IgG and IgA properly. And their lymph nodes are impressively abnormal. Any pathologist that looks at these lymph nodes, they go, they get worried, they get scared, and then they send the biopsies for second opinion to NIH. And our pathologist calls us and says, there's another PI3 kinase patient, we think, here. We have the slides. Do you want to give a call to the physician? So that's how we recruit these patients, basically, because of our pathology department. And what you are seeing here is that you cannot see it in organized, nice germinal centers, follicles. It's all big mess. And the brown stains here is the T cells. So in the germinal centers, in the follicle centers, you need to have these T cells that are follicular helper T cells. But in the case of the patients, it's full of disorganized T cells. So that's a bad look. And when we do a PET scan, their lymph nodes lit up. And what you're looking at here is the pre-treatment PET CT of one of the patients. Has multiple, actually this is the patient that Dr. Alpan has referred to us with big liver here and big spleen with multiple areas of increased activity. And after rapamycin treatment, not only those areas have disappeared, but the size of the liver and spleen has shrunk impressively. And this is the CT scan images of the of another patient. So we have a disease, but we also have a treatment that interferes with the pathway that is overactivated due to this mutation. So the other side of the story is that this disease leads to EBV, susceptibility to EBV infection, and therefore lymphoma. Most of the deaths in these families that we have seen have been due to lymphoma and aggressive lymphoma. So another option for these patients is to go with the bone marrow transplant early on and identify these patients, screen their families early on. And the last defect that I know I'm running out of time probably is I will mention shortly because we just published this earlier, last month, not this month, last month. And it is one of those diseases that we always heard in the immunodeficiency community, but didn't know about the gene much. So Mike Nelson told you about the common variable immunodeficiency and granulomas in these patients having sometimes lymph nodes or not. So we had the opportunity to see this patient a few years ago. She was 10 years ago. She was 9 years old at that time, and she was referred to us for a second opinion. It took us 10 years to figure out her mutation, her defect, but it led to the discovery of this genetic defect in other patients with a similar problem. So she had initially ITP at age 2. I don't know how many of you see little toddlers with ITP, but this led to was followed by nodules in the lungs and brain because she had seizure and had this inflammatory non-caseating granulomas in the lungs. She had low immunoglobulins. She was lymphopenic. Had many genes looked at. People tried to figure out what she had exactly. One thing they did not do that worked for us, I don't know if it worked for her, was transplanting her without a genetic diagnosis. So she had her gut infiltrating with lymphocytes. You don't see this, and you don't see these kind of brain lesions, that's inflammatory lesions, without too much neurological sequelae or deficit. Then several years later, his father called us. I said, well, I just got diagnosed with common variable immunodeficiency. My immunoglobulins are low, and because I have had a couple of pneumonia sinus infections. So that was a big deal for us, having two members of the family probably with the same genetic defect. And suggests a dominant inheritance. So we wanted to see him, and we saw this patient. He had the similar inflammatory lung lesions and some bronchiectasis. He had thrombocytopenia, like his daughter, but he had what we call LGL leukemia. It's large granular lymphocytes becoming clonal and causing suppression in the bone marrow. And then he also developed neuropathy. So this is what you're looking at, his gut biopsy. That huge, big brown stain there is a cluster of B cells at a place where they are not supposed to be. So lymphocytes are trying to form germinal center in intestinal mucosa where they should not be doing that. So we looked at these patients' phenotypes, and we figured out that they had CD4 lymphopenia as well, lost their naive T cells. They had progressive loss of B cells. What you're seeing here is lack of B cells. And then they over time, their B cells, because we were able to follow this girl for around 10 years, progressively decreased, and they had low immunoglobulin production. So when we did this whole exome, we found a deleterious mutation meaning causing a stop.
May I have your attention, please? May I have your attention, please? A fire emergency has been reported in the building. Please leave.
I must have gone way over time. Okay. Are we supposed to leave or? You're okay? All right. 2 minutes. I'll finish the story in 2 minutes. But this family, the daughter and the father, in whole exome sequencing, we found a deleterious mutation at the beginning of exon 2. This is in this gene, and the same protein encodes for the same protein called CTLA4. It's a big deal because CTLA4 is one of the well-known receptors in the world of immunology. It is heavily involved in regulation and autoimmunity, and they have even designed a blocker or a fusion protein that mimics CTLA4. Have you heard of Orencia that's used in rheumatology? So Orencia is what would ideally replace CTLA4. It's a soluble version of CTLA4. So that's why this molecule is very well studied, understood, and it was a shock to us that nobody has seen this before, but we did. We did stain for the protein. That's what you're looking at here. CD162 is the other name for CTLA4. And we saw diminished, not absent, because this is a heterozygous mutation. 2 alleles, one of the 2 alleles is wild type. So you're looking at diminished expression of CD152 in the patient, and that's normal. The gray one is the MAC control, isotype control. So what this thing does is really gets expressed on regulatory T cells. So it tells the other cells, effector cells or antigen-presenting cells to stop. Because it binds to its target 8086, and it dislocates this thing called CD28 and stops dendritic cells from showing the antigen to the non-regulatory, non-Tregs. And when it's absent on regulatory T cells, this whole process of antigen induced proliferation of effectors and antigen being recognized keeps going on. So you end up with autoimmunity because there's so much antigen recognition, self-antigen recognition by the effector cells. And there's overproliferation because if the cells don't have CTLA4, they don't stop proliferating. Their threshold for activation is very diminished. So we don't have the normal death at a timely manner. So we have found now it's 4, this is our original paper that we had 4 families. Now we are up to 8 families with multiple generations having common variable immunodeficiency. But interestingly, back in 60s or 70s, their grandmas or moms were diagnosed with celiac disease. It's celiac disease because what we see in the pathology is transepithelial T cells, lymphocytes, and that's something that you do see in celiac disease. So I think some of the patients that were initially diagnosed with celiac and ended up having common variable will probably end up having this mutation, this genetic defect. But the clinical presentation is quite interesting. Every single patient had neuropathy, like diarrhea, or was diagnosed with celiac disease elsewhere. They had inflammations in the lung and brain, these inflammatory infiltrates, and some of them were common variable. Not every patient had common variable immunodeficiency, and one of the patients and one of the families, I think he is patient C, was diagnosed with lymphoma at age 10. So it's also predisposing to lymphomas.
May I have your attention, please? May I have your attention, please? At this time, we have investigated the source of the alarm and have determined it's safe to occupy the building. While you may see strobe lights for a few more minutes, we in the fire department have determined that there is no cause for alarm. We apologize for your inconvenience and thank you for your understanding.
Lo and behold, we have shown that diminished CTLA4 expression in the regulatory T cells. So it turns out that this is like a disease, very much like IPEX that you have heard of. CTLA4 affects regulatory T cell function, and you end up with all this autoimmunity. We did lots of studies looking at Treg function, these cells being able to inhibit effector cells, and that's what we showed, that they had functional problems. I am showing you the brain biopsy of one of these patients. So it's an impressive disease. Some of the patients have these diffuse infiltrates in the brain with a lot of edema, yet they're not as symptomatic as you would think they would be. And they end up having these granulomas in the brain that are made of T and B cells. But if the patient have received rituximab, anti-CD20 treatment for cytopenias, they come to us with clonal plasma cell infiltrates. So that's what rituximab does, changes the biology of the pathology of the organ infiltrate. And then pathologists looking at the second biopsy get scared about a low degree B cell lymphoma. So we have identified these mutations in CTLA4. It's actually leading to haploinsufficiency. We have multiple families with affecting multiple generations. And we decided, we were able to tell the story that CTLA4 is indispensable for the homeostasis and immune regulation. And these findings are in contrast to the mice, because mice this gene was knocked out in mice, and mice heterozygous for this defect didn't have a clinical phenotype. So it tells you that in humans, these genes that control, regulate the immune system are much more tightly controlled. So you need both alleles to be in great control. So that's quantity, like 50% or less would lead to a lymphoproliferative phenotype with lymphadenopathy, but some splenomegaly, but mostly lymphocytic infiltrates in the organs. So these are the people that have done a huge amount of work for all these 3 gene discoveries, but mostly none of this could have been done with the person in the center, Julia Niemela, who's our bioinformatics person. So whole exome sequencing is only central if it's very central, but it's only meaningful if you know what you're looking for. And to know what you're looking for, you have to have a good bioinformatician who will go through the genes, who will look at the scoring and help you figure out what is the one that you want to go after. And all the other people are the people who really put a lot of work into this project. That's all.